Welcome, everyone. I'm Rachel Swanek. I am the Northeast Program Manager with the Forest Stewards Guild and the manager of the Sustaining Ash Partners Network. I want to welcome you all to our kickoff event for the Pathways to Sustain Ash series. This is a three-part series that will explore ash seed collection from sourcing to collection to propagation. We've got two more events coming up after this one. So we have another webinar on March 6th at 1 p.m. It's going to be a deep dive into the how-to of ash seed collection, and then an in-person field tour at the New Hampshire State Nursery on March 20th. It's going to be happening at 9 a.m. So we'll put some links into the chat for upcoming events that are part of this series, but very excited to have you all here for our first event. So to get us started, I'm going to share a little bit of background about the Sustaining Ash Partners Network, and I'm also going to share land acknowledgement. Share my screen. All right, does that look okay for folks? All right, wonderful. So the lands at the core of our network and that many of us presently inhabit in the Northeast sit on the ancestral homelands and waterways of the region's indigenous peoples. We acknowledge that these peoples have been stewards of these lands since time immemorial and are grateful for their continued stewardship and care of this place. For our region's tribal nations, issues of sovereignty, territorial rights, and access to sacred sites are ongoing. We acknowledge that there are inherent rights to sovereignty, and we also recognize the painful impacts of settler colonialism that attempted to remove tribes from their homelands and erase their cultures. SAP-Ne is committed to building cross-cultural connections that celebrate the vibrancy, knowledge, and adaptability of our region's tribal nations. So with that, I want to share a little bit of background about what SAP-Ne is for those that aren't familiar with the network. SAP-Ne is a regional network led by the Forest Stewards Guild and our partners who work across tribal nations, state and federal agencies, NGOs, universities, all across the Northeast. I've listed some of our partners here. You can also find a full list of our partners on our website. We were founded in response to the impacts of the non-native pest, the emerald ash borer, which threatens to eliminate ash from our landscape. Our efforts at SAP-Ne focus on both the culturally important brown or black ash, as well as other ash species that are found in the region. And what we're seeing presently is that our attention is shifting from limiting spread to responding to more active infestations of EAB. And given this, a unified approach around stewarding ash is really more important now than ever if we want to sustain ash on the landscape. SAP-Ne aims to support collaborative stewardship by building off ongoing efforts around ash that are already happening in the region and also helping to develop more cross-boundary partnerships. Some of our objectives are to encourage stewardship approaches that honor and amplify the cultural values of ash, to offer workshops and webinars like the one that you're attending here today, to establish a network of demonstration areas in the Northeast that will highlight ash stewardship, and to develop a web hub to share resources on ash. Our ultimate goal is to support a community of partners across the region who are committed to keeping ash on the landscape for future generations. And we're excited that you're all here to be part of our community. If you want to find out more about our network, you can check out our website and you can also contact me. My email's up here. Happy to talk to folks who want to get more involved on the organizing side or want to hear about events or make suggestions for ways that the network could support the community. Just gonna grab that. All right. So with that, a little bit of background about SAP-Ne. We're going to do a quick poll of attendees. This is sort of part of our funding source that we track participant time at events. So we're going to get that launched. And if you all could just provide a little bit of information about how your time is being covered, that would be much appreciated. And while you're doing that, I'm going to share just some ground rules for the meeting. So the webinar is going to last 90 minutes. We ask that you keep yourself muted during the webinar unless you're kind of called on or you're asking a question. There will be time for Q&A. The webinar is going to be broken up into two different sections. So we're going to have one initial talk, and there'll be a Q&A after that. So if you want to ask questions, you can put them in the chat during the talk, or you can save your question, use the raise hand function when it's time for the Q&A. And then we'll have another part of the talk. We'll have 3 talks concurrently after one another. And then there'll be time for Q&A after those 3 talks. But the survey disappeared. Let's try maybe launch it again. All right. Okay, wonderful. I'll give everyone like 30 seconds to quickly get their answers in, and then we'll go right into the first talk. All right, wonderful. So let's go right ahead and get into the webinar. Our first speaker is Dr. Nate Seigert. Nate is a forest entomologist with the U.S. Forest Service who has studied and tracked EAB since the start of its North American invasion, originating in Michigan in the early 2000s. He is broadly interested in the ecological and evolutionary aspects of plant-insect interactions, population dynamics of forest insects, and invasive species biology. Nate has been a leader in efforts to understand and respond to the spread of EAB, sharing technical assistance, research findings, and facilitating cross-boundary collaborations with managers and landowners throughout the region. Nate, do you want to take it away?
Sure. Thanks a lot, Rachel. Let me just share my screen here. All right, did that come up for everybody? All right, excellent. All right, yeah, so as Rachel said, I'm a forest entomologist with the Forest Service based out of Durham, New Hampshire. Um, and I've been working on emerald ash borer for quite a while, just getting this screen straight here. Um, and Yeah, I'm happy to be here today. So for context, emerald ash borer is the most destructive and costly forest insect to invade North America. Um, Not just in recent history, but ever. It's killed millions of ash trees already, and it continues to do so at an alarming rate. Um, so emerald ash borer on its own doesn't spread very far. It's usually less than a quarter to a half mile a year through the flight of the adult insect. But it is readily moved by humans tens or hundreds of miles to new areas and infested firewood or other ash materials. And it's really this short distance and long distance dispersal mechanisms, along with the remarkable reproductive potential of emerald ash borer, that's facilitated its invasion in North America. So invasion of EAB is characterized by having a kind of a core infested area surrounded by isolated satellite infestations some distance from the generally infested area. So over the last 20 years or so, the infested area has continued to expand as we've detected established populations of emerald ash borer across much of the east, and more recently on the west coast in Oregon and British Columbia. When we examine invasion dynamics at regional scales, such as the northeastern US, we see similar patterns. New and isolated satellite infestations become established each year, and older satellite infestations continue to expand and begin to coalesce with other nearby satellite infestations until large areas become generally infested. And the same holds true for invasion dynamics at local scales. This is some tree ring work that I did. It's reconstructed the establishment and spread of emerald ash borer in North America in southeastern lower Michigan. And again, you can see kind of that core infested area surrounded by isolated satellite populations. And then over time, they just coalesce and they kind of all clump together. So when we evaluate host susceptibility to emerald ash borer, we consistently find that our North American species of ash are more susceptible than Asian species of ash, and European species of ash tend to be more intermediate in susceptibility. And even among our North American species of ash, we see a range of susceptibility to emerald ash borer. Black ash is our most vulnerable species, whereas blue ash tends to be more tolerant to emerald ash borer infestation. Other species like white ash or green ash fall somewhere in the middle. Um, and you'll see this this gradient here has a lot of red even through the middle, and you'll see some of that why I have that because it's not evenly distributed across this gradient. Emerald ash borer can basically kill all susceptible trees greater than 1 inch in diameter, and trees may only persist through either vegetative stump sprouts as the main tree dies, or through seed production. So collecting and preserving ash seed is a useful step to take because the existing seed bank in natural systems doesn't stay viable for many years, and emerald ash borer can quickly kill seed-producing overstory trees, leaving us with an orphaned cohort of ash regeneration. I say orphaned because it's kind of left all on its own. There are no seed-producing trees above it, and there are no newly germinating seedlings below because we don't have a seed bank anymore. So we have three primary ash species in the Northeast: black ash, green ash, and white ash. And they've all been classified as being critically endangered due to the emerald ash borer invasion. And I'm going to talk about each one here in a little bit more detail. So black ash is the most vulnerable to emerald ash borer, and it is a cultural keystone species to many tribal nations in the US and First Nations in Canada, with basketry traditions in Eastern North America. And in post-invasion forests, infested black ash often attempt to produce stump sprouts as trees die, but studies have shown that none of those stump sprouts survive after more than a few years. And we lose black ash overstory trees very quickly in infested areas. So here's some data from a mixed black ash and green ash forest in Michigan, and you can see that 100% of the black ash overstory died within just a few years of one another. And some more recent work shows that emerald ash borer has been established long enough across much of the lower Great Lakes region that heavy black ash basal area losses have occurred already, more than 95%, which is virtually every single black ash stem except for maybe the smallest seedlings and saplings. And combining emerald ash borer spread with how quickly we know it kills ash suggests that by 2035, much of the range of black ash is likely to be incurring more than 75 to 95% basal area losses across the Northeast and beyond. So switching to green ash is also pretty highly vulnerable to emerald ash borer infestation and decline. In post-invasion forests, we found that 25 to 30% of the EAB killed green ash have surviving stump sprouts after about a decade of EAB being there. But their long-term survival is unknown. They're still, these stump sprouts are still small in diameter. A lot of them are smaller than the width of my pinky. And they have a lot of characteristics similar to young saplings, like smooth bark, which emerald ash borer don't like to lay their eggs on, really smooth bark trees and things of that size. So they're surviving, at least 25 to 30% of them. And here's the other half of that data that I showed from the mixed black ash and green ash forest in Michigan. You can see that 100% of the green ash overstory also died, but it took a couple of years longer than our super susceptible black ash. Here's just another example for green ash. This is a different site in Michigan where we had inventoried all the green ash before emerald ash borer was present. And this is the metrics in ash phloem area, but it gives you a sense of how much ash was there before EAB entered the picture. And this is the difference 10 years after EAB arrived. You can see like even the highest size class here barely has any ash phloem left in it. And if you calculate the basal area losses at this site, we have more than 94% basal area losses of the green ash after 10 years. So white ash is considered intermediate in susceptibility to emerald ash borer and has shown some tolerance in areas where emerald ash borer has been established the longest. So 75% of the total stems were estimated to be alive in one study, but it was highly variable from site to site. So some sites approached 100% survival, while others had no survival whatsoever. And in this photo is an infested ash tree see if I can use the pointer here. Yeah. So there's an infested ash tree kind of on the left side that has a really thin crown, and the tree to the right of it um, is a white ash that appears to be unaffected. So we're seeing some white ash trees being killed readily by emerald ash borer, while others are remaining on the landscape. So research is underway to determine what factors may be actually influencing this. That's promising, but it's important to note that this hasn't been observed in the Northeast yet, and we continue to lose many overstory white ash trees. So here's some data from an infested forest with black ash and white ash that I've been assessing annually for several years in New Hampshire. You can see the black ash was impacted quite quickly, whereas the white ash started off pretty well, but it's continuing to die from emerald ash borer. So as of August of last year, 2024, I'm down to only 8 live black ash trees, and they're averaging about 2.5 inches in diameter. So the orange part of each one of these bars are the dead trees, and the blue part is what's still alive. So you can see that there are just 8 remaining black ash trees in that small size class. Those 8 surviving trees, one has the healthy crown and the rest have a range of canopy dieback. So they're declining, and we're probably going to continue to see losses over the next couple of years. For the white ash, I started out with a lot more trees at that site, but I'm down to 32 trees that are alive right now. And they range across all but the largest size class. So you can see heavy tolls in the 18 to 23 inch, the 11 to 17 inch, and the 6 to 10 inch diameter classes for white ash. Like black ash, they actually have a very similar distribution of canopy dieback. So whether our eastern ash species will function as overstory trees in post-invasion forests in North America, it may be jeopardized by this loss of seed sources as we lose overstory trees, and also by the endemic EAB populations that are present that likely will limit recruitment of existing ash seedlings and saplings. But as bad as that is, opportunities to preserve ash remain. So this data right here is from the Forest Inventory and Analysis, and it shows total basal area per county of white ash and green ash combined in the Northeast. And as you can see, we currently have a lot of ash still remaining and still producing seed across the Northeast. So as such, I think this is a very timely webinar, and I'm happy to see so much interest. And with that, thank you. And if there's time for questions, I can take some of those.
Nate. If folks in the group have questions, you can put them in the chat or you can use the raise hand function on Zoom.
Nate, they just moved the quarantine in northern Maine down another 25, 30 miles. Would it be wise to quarantine the entire county?
Well, yeah, I mean, it's a challenge. I always view quarantines as one of our management tools. So it's usually useful early on in an infestation when we can kind of limit people moving infested wood long distances. But it's a balance between being able to facilitate moving wood and limiting the spread of emerald ash borer. So I mean, there's probably a lot of pros and cons, Richard, whether it's worth extending it across the whole county. We know if we do that too early, it really just facilitates the spread of emerald ash borer. So
Yeah.
I would say starting off, it probably makes sense to limit the quarantine as much as possible, but there's a balance there.
Yeah, well, the latest move on the quarantine is between me and where I get my basket wood. So,
Yeah,
so I can't go up there and get the basket wood and move it down below the
right.
quarantine.
Yeah, that's a perfect example of the balance, right? Like now you'd be forced to process it in the quarantine area to make it safe to move.
We had a couple questions in the chat. One from Jenny Tibbetts. I've read about monitoring of ash trees using technology like drones. Is the USDA doing some of that?
Yeah, we monitor and survey for emerald ash borer in a lot of different ways. Oftentimes with state and private partners, and you know other forest health specialists. So yeah, we use a range of technologies, everything from just visual observation to drones to using traps and girdled trees, which are really attractive to dispersing EAB. So yeah, we try to use a mix of technologies in order to get the best information we can.
Another question from Judy Fulton. How much does emerald ash borer persist after 100% of mortality in the ash trees?
Yeah, that's a good question. So we lose 100% of the overstory trees pretty readily, but we usually have enough ash hanging around that's in some sort of decline, like oftentimes smaller diameter trees, that we haven't seen sites where emerald ash borer killed all the trees and then died out. So there's usually some low level population of emerald ash borer that persists so that unfortunately, when those surviving saplings start to be released from, the overstory opening up and gaps being formed, and they start to put on growth, they quickly grow into size classes that readily get attacked by emerald ash borer again. So yeah, they don't disappear like we were hoping they would for a while.
Another question about give like a timeline on what's the timeline for green ash? How long will it be killed once EAB infestation gets into an area?
Yeah, so generally what we've been finding is, especially with green ash, like we have most of our overstory trees in a stand will be killed by emerald ash borer within 8 to 10 years of it being introduced into a stand. Um, so that's where you see the heaviest losses, and then you kind of switch into those post-invasion forest dynamics where you might have seedlings and saplings that are growing into larger size classes. But it's that first, you know, 8 to 10 years is what's the most critical.
Question from Donald Campbell. Will you repeat how small a diameter will experience full mortality from EAB?
Yeah, so small diameter, I think anything less than 1 inch is pretty safe. So we'll have that smallest size class present. It's just that when it's released from this gap formation, it grows quickly into larger size classes that makes it very vulnerable. And the issue is, of course, if we don't have seed producing trees anymore that are laying ash seed down, then all we have left is that one cohort of ash seedlings and saplings present. So we're trying in a lot of areas, we're trying to protect overstory seed producing trees through chemical injection and other tools so that we at least maintain seed coming down in certain areas. But it's pretty limited compared to the whole range of ash across the Northeast.
I'm gonna have one more question, and then there's a few more in the chat. Maybe Nate, you can respond in the chat after. Um, Tina asks, I have many green ash seedlings coming up in my yard, around 3 to 4 years growth. What are your suggestions that I should do for managing?
Yeah, I'm in the same situation. I've got some white ash that I've actually been growing in my yard as well, and in they're growing like crazy right now because ash still is a great tree. Um, So I would monitor those, and as they get into like a 1 or 2 inch size class, really monitor to look to see if you've got emerald ash borer on any of the trees that are in the area. And if you want to keep them, there are certainly tools like chemical and insecticides that are available that do a really great job protecting individual trees. So if you've got a tree in your yard and you want to protect it, there are certainly ways to do that.
Nate, can you inject a tree after the emerald ash borer has gotten into it?
Yeah, Richard, you can. You just have, usually we advise that the canopy hasn't had a lot of decline yet.
Okay.
So usually it's, I don't like injecting trees necessarily that or have more than 25% canopy decline. And that's just because if you have too much canopy decline, that means that there's so much EAB already in there that it
Yeah.
disrupts the translocation of the insecticide in the tree, and it doesn't work. So you want to inject those while there's still the ability for the tree to take up that chemical and protect it.
Okay. Thank you.
Yep.
All right, thanks so much, Nate. We're going to move on to the next part of our webinar. I saw the last question that came in was about seed repositories, so that's a great transition. I think we're going to get into that in this next portion. So we've got 3 speakers. I'm going to introduce our first speaker. So Jessica Raspitha is the environmental specialist with the St. Regis Mohawk Tribe. Jessica is a member of the St. Regis Mohawk Tribe and has worked full-time for the tribe since 2014. The St. Regis Mohawk Tribe has been a leader in the region in promoting forward-thinking strategies to steward ash for decades. Jessica's work has included promoting integrated pest management strategies, helping to start the tribe's native plant nursery, and developing strategies to battle EAB infestations, which threaten the forest ecology in their territories of Akwesasne and the cultural practices of basket making. Jessica?
Hi, good morning, everyone. So let me share my screen. Okay, is everyone seeing that?
Yep.
Okay, perfect. So yes, my name is Jessica Raspitha. I'm the Land Resources Program Manager for the St. Regis Mohawk Tribes Environment Division. And so today I just wanted to provide some insight on the way that we prioritize ash seeds in some of our ongoing restoration work. I'm super appreciative to have been invited to speak, and even more appreciative to see how many people have joined and that there's this much interest in ash seed preservation. Okay, so to provide some background, the Land Resources Program is one of 6 environmental programs within the St. Regis Mohawk Tribes Environment Division. The Land Resources Program is the newest, which was formed in 2019. And the intent was to create a cohesive team to oversee our various land-based restoration efforts, which include forestry, invasive terrestrial species, oversight of our pesticides program and tribal pesticides permitting process, and the operations of our native plant nursery. And I think it's important to reference the different branches within our team, specifically because there's a lot of overlap in our efforts. And the way that we mitigate damage caused by the emerald ash borer, it really is a whole team effort. So central to our mission, to the mission of our program, is the acknowledgement that nearly all of our cultural practices are tied to our natural resources. And for those practices to continue, those resources need to be both available and safe to use. Without going into our community's very long and complex history of industrial contamination, I did want to express that cultural practices and resource use vary greatly among individuals. But today we're going to focus specifically on black ash and the impacts that the emerald ash borer is having on this cultural resource. So I'm sure many of you are very familiar with the emerald ash borer, but just in case anybody is not, the EAB is an invasive beetle that was first found in the US in 2002, and it has seen nearly a 100% mortality rate among ash trees. And this is particularly problematic for us with EAB first being found within our tribal territory in 2016, because ash is one of our dominant species in our landscape. And so we're not only losing ash as a cultural resource, but the dead standing trees left behind pose a public safety hazard, especially when these trees are located along roadways, power lines, parks, and buildings. So before we get into cultural use, I think it's important that we understand the very specific way that an EAB kills a tree. So part of the EAB life cycle is that the beetle will lay its eggs on the bark of an ash tree, and the resulting larvae will then bore into the vascular tissues of the tree. And unlike other wood-boring beetles, the EAB makes these very characteristic meandering S-shaped galleries as it grows and moves through the tree. Um, and ultimately, this impacts the tree's ability to move water and nutrients from the roots up through the canopy, and it eventually kills the tree. So to show what this damage looks like, the photo on the left are new galleries, and you can actually see there's still a live larvae in there. And then the photo in the middle, this is an older gallery where the tree has attempted to protect itself by scarring over that tissue. And from the discoloration, you can see that that vascular tissue is no longer viable, and that part of the tree is no longer able to move nutrients. And this is very unfortunate for us, specifically because these inner bark tissues are the same part of the tree that we use to create black ash splints as part of our traditional basketry. So splint is obtained by peeling back the bark of an ash tree, specifically black ash, and pounding the log in a way that separates those annual growth rings into the fibers that we use for our baskets. And I did attempt to include a video, so we will play that. So this is Louis Gray. He's one of our community's elders and well-known basket makers. And I wanted to include this video demonstration because making splint is a very skillful and labor-intensive art. And while Louis makes it look very easy, I can assure you that it's not. And this practice, along with all of the other traditional ecological knowledge that comes along with it, is passed down through generations. And so to elaborate on that, the traditional ecological knowledge tied to basket making isn't just this technique, but it's also how do we identify which tree in the forest will be ideal for baskets, because not every black ash can become a basket. When is the correct time to harvest a tree? How do we make the splint? How do we clean the splint? How do we store that splint? And then how do we make the basket? So it's really important for us to make sure that we still have ash as a resource, because we can't continue to pass those traditions down and that knowledge down without having access to the resource. So the end result of the splint making, the tree harvesting, are the baskets that we make of black ash splint. And in Akwesasne, we primarily make utility baskets and fancy baskets. And I also want to note that black ash basketry is not specific to the Mohawks. There's many tribes in the Great Lakes region and the eastern U.S. With very similar practices that are also working to mitigate the impacts of EAB for the same purpose. So there's a couple things that I want to note in these pictures. First is that there are other native species that are often incorporated into baskets. Specifically, this first photo on the left, this is a sweetgrass fancy basket. And it has sweetgrass woven between the black ash splints. And I particularly like this photo because this basket was made using a braid of sweetgrass that took 50 yards of sweetgrass to weave. And then we took this picture at our native plant nursery in our sweetgrass patch. And I also wanted to acknowledge that we also have photographed as a member of our team, Angelo Johnson. He happens to be a 6th generation basket maker. Angelo is our lead field technician for black ash and EAB work, and having him on our team and in the field assisting with our management decisions is how we're able to ensure that our cultural context is being incorporated. So I also wanted to touch on the function of some of these utility baskets, like the ones Angelo makes, and how they relate to other cultural practices. So the Haudenosaunee people are known for our cultivation of corns, beans, and squash. And to this day, the St. Regis Mohawk Tribe's agricultural program remains a large producer of Haudenosaunee white corn. White corn it's a large part of our traditional diet, but it's not quite like the corn that you would find at a grocery store. It's much larger, and the kernels are bigger. And notably, the kernels also need to be washed before they can be eaten. And what that means is that each hull of each individual kernel has to be removed. And the best way to do that is with a corn washing basket. So as you can see in the picture on the left, there's a process involved in washing corn, and the cooking process helps to soften that hull but the hulls still need to be manually removed from each seed, from each kernel. And so there is a utility basket that has a particular weave that makes the inside rough so that rather than having to remove those individual hulls by hand, you can process a large amount of corn using that basket. And to this day, our agriculture program uses corn washing baskets when they harvest at the end of their season, and they process all of their corn before it can be eaten or preserved or ground into flour. And I wanted to mention this function because while we talk about the implications of EAB and what it means for basketry, these impacts are further reaching than just baskets because we also have a lot of function in those baskets that are tied to other cultural practices and ceremony. So there are a number of efforts being taken each year to mitigate the impacts of emerald ash borer. And while we could spend all day on this topic alone, I wanted to just run through them really quick. So each year we're involved in monitoring for EAB in partnership with USDA. And this is just to help monitor the movements and trends of our local EAB population. We also treat a select number of trees using an injected pesticide that provides up to 3 years of EAB resistance. We also work with the USDA Biocontrol Lab to release parasitoid wasps in black ash stands that have known EAB populations in an attempt to foster populations of wasps that can predate on EAB eggs and larvae. We are also involved in silvicultural treatments, mainly in partnership with the New York State Department of Environmental Conservation, working in nearby state forests to help remove some older growth from these black ash stands to open up the canopy and promote regeneration habitat. And we are also selectively harvesting basket grade trees so that they can be processed into splint before the trees are impacted by EAB so that we can preserve its cultural value. And we also undergo a tree planting effort each year to try to replace some of the canopy that's lost to the trees that do succumb to EAB damage. And we are lastly collecting ash seeds. So I wanted to elaborate a little bit more on the pesticide injection since it's closely related to our seed collection efforts, and that one of the criteria that we use for selecting trees for treatment is that they have basket grade qualities. So not all trees can be injected. They need to be of a certain size and they need to be in good health in order to properly uptake the pesticide that we use. And because the goal of the pesticide is that the EAB won't be able to bore into the tree, we also want to make sure that the tree is in good health, like Nate had mentioned. So because injecting trees can be a lengthy process, because the products that we're using are fairly expensive, and because each injection only provides 3 years of protection, we need to be really selective on which trees we are choosing to protect this way. So just to specify, we're using an ArborJet product and Triage. It's amamectin benzoate product for the injection. And because of that 3-year cycle, we need to revisit each one of these trees on a recurring 3-year cycle to maintain that protection. And so while we're being selective, what we're looking specifically for to determine whether or not we're going to treat a tree is whether or not it's occupying ideal habitat, whether or not it's seed bearing, and whether or not that tree has basket grade qualities, because we want to try to preserve those qualities through the genetics of our collected seeds. Okay, so as part of our annual seed collection efforts, we've been largely guided through our working relationship with the Mid-Atlantic Regional Seed Bank, or MARSB, and they've helped us understand the required genetic diversity needed to properly support our nursery operations. So for each species that we propagate through our plant nursery, we're ideally looking to source from about 50 individual seed sourcing locations, and this requires a lot of field reconnaissance work and identifying which areas are ideal for seed collecting. It requires a lot of revisits to those sites to make sure we're properly timing the seed collection. And then it also requires our cultural consideration. And we have here Angelo explaining what specifically he's looking for when he decides a tree is basket grade or not. He explains the bark texture, the size, how far off the ground we go up until we get to branches, and like just looking around at the different growing conditions to say whether or not he believes that this tree will create good splint. And so in response to our emerald ash borer infestation and the cultural significance that ash trees have for us, we have an Akwesasne Mohawk territory emerald ash borer community response plan that was created in partnership by the St. Regis Mohawk Tribe, USDA, and also the Akwesasne Task Force on the Environment. And the intent of this plan was to lay out our plan of action for how we would protect ash as a cultural resource, how we're going to promote the longevity of the practices tied to basketry, to outline how we will carry out our mitigation efforts, and how we will ensure that seeds are collected to ensure the longevity of the species. And so we've been following this plan since our program was formed, and even more so with the new operation of our native plant nursery beginning in 2021. And so the seeds that we collect, they're collected by our plant nursery technicians, and they are jointly stored both internally within our own warehouse and also in cold storage with the Mid-Atlantic Regional Seed Bank. And we've also shared some of the collected seeds with other tribal nations who have asked for them. And so our long-term restoration goals moving forward include continuing to remove the hazard trees that they grow in number each year, replanting trees to try to restore that lost canopy and that lost ecological function that all of those trees were serving for many years, and also continuing to propagate trees through our native plant nursery, trees that are not susceptible to EAB but also native to our area. And while we aren't currently propagating ash because we haven't reached the peak of our infestation yet, and it's still a little early, we are preparing what our nursery operations will look like and making those plans so that someday we can start ash propagation and hopefully start putting ash back into our post-EAB landscape in the future. Thank you.
Thank you so much, Jessica. And if people have questions, we're going to do them at the end of the 3 talks, but you can certainly put them in the chat now if they come up. All right, we're going to move to our next speaker. It's Dr. Jill Hamilton. Jill is an associate professor in ecosystem science and management and director of the Schatz Center at Penn State University. Jill is a leading researcher whose work aims to quantify and preserve genetic diversity across ash species to help develop breeding and reforestation programs for climate and EAB-resistant trees. Jill leads the growing Hamilton Lab for Plant Evolutionary and Ecological Genetics over at Penn State. Jill?
Thanks so much, Rachel. And thanks everybody for being here today. And thank you for the invitation. It's a real pleasure to be here to talk to you about a species that we're quite passionate about here in Pennsylvania, and to kind of provide a perspective of the research that we do at Penn State, where we're taking a genetics approach to conserving ash species writ large across North America. And as part of this goal, we want to create a molecular passport for our collections that we're very actively involved on to enhance monitoring and conservation long-term for Fraxinus. And the reason that we focus on thinking about genetics and the molecular passport in particular is that an overarching goal of our research is really to maintain that ability, technical issues, to maintain an ability to cope with abiotic or biotic threats that are impacting our native species. And we think about this a lot in terms of maintaining species adaptive evolutionary potential. So to conserve that genetic variation that is standing on the landscape today. And we think about this particularly in terms of how we design and implement our conservation collections, how we transfer seedlings or seeds that we use for restoration, and how we approach the conservation and restoration of our rare and endangered species, where ultimately, as geneticists, the questions that and approaches that we need for conservation and restoration of our forest trees really requires this interdisciplinary approach spanning both lab work, field work, and data science. And so today, I thought I'd talk about the different approaches that we're thinking about for optimizing preservation of evolutionary potential in our ex situ collections, and to indicate what the role of genetics is in the collection itself. An ex situ collection often aims to capture 95% of a species' genetic diversity. The IUCN recommends capturing 95% of that genetic diversity on the landscape today to ensure that genetic variation is maintained to enable species to respond to changing biotic and abiotic threats in the future. So I'll talk a little bit about the ex situ collections themselves and how we can leverage information within the collections today to understand the distribution of genetic variation in our populations, in our collections right now. And then I'll also talk about opportunities where we can enhance the information that is available within our current collections by pairing them with genomic data to create, in addition to our traditional passport data that we have associated with our ex situ collections, a molecular passport to understand the genetic variation of those maternal trees that we're collecting on the landscape and create a molecular passport to complement our seed collections to understand the genetic variation on the landscape today. Okay, so the first thing when doing the collections, as has been mentioned previously, the key to these collections, they are a lot of work and they're quite expensive. The key is collaborative partnerships. And we've really benefited from working with our state, national agencies, including the National Tree Seed Center in Canada, the U.S. National Plant Germplasm system in the United States, the U.S. Forest Service, and many volunteers that have gone out and captured these collections. And these collections provide us with a snapshot of that variation associated with genetic variation at that time and in that place. So each collection that we have reflects that very time, that time in that environment for that individual. And so when we're thinking about collections, it's important not to only think about a one-time collection, but the added advantage of having temporal sampling to create a legacy of variation that occurs over time within our collections. So as part of this project, we aimed to work with our partners to preserve the genetic library of black ash across the whole range of black ash, including many populations in Canada and the US, and to assess the genetic variation that we actually had preserved in the collection today. And so one of the tools that we wanted to use to assess this was seed morphology, where we can take our seed collections and actually take x-rays of collections of our seed collections and assess morphological variation for traits associated with our seed collections. And this is work that was spearheaded by a former postdoc, Melissa Lear, and an amazing undergrad, Lucy Anderson, where we leveraged collections of black ash seed from across the entire range of black ash. And we had about 100 seed that we were working with. And we with our U.S. Forest Service partners, we x-rayed about 50 seed lots, seed individuals per seed lot, and we covered the entire range of black ash that was available at that time. And you can imagine that for an undergrad, this is a really exciting project because you get into using ImageJ and you start measuring traits associated with wing variation and seed variation to see how that variability in the morphology of the seed is partitioned across this broad landscape in which these seeds persist. And we know that morphology can be a reasonable trait to use as a proxy for genetic variation because it is largely heritable from parent to progeny. And therefore, we can leverage this foundational knowledge in the morphology of our seeds as a proxy to estimate and understand the standing distribution of genetic variation in our ash populations today, even if we don't have genomic data. Now, we had about 9,000, many thousands of ash seeds for this collection. The challenge is doing all these things by hand. So we were very lucky to work with an amazing PhD student, Dulzadma Devassaran, alongside Kirilo Piccolo, who have now developed a machine learning approach to segment images that were x-rayed for these ash seedlings. And this machine learning approach allows us to quickly assess all 50 seeds within these seed lots for morphological differences associated with both the samara and the seed. And so you can see in this image here, how we start with our X-ray on the left and how we can divide and assess both variation that we observe in the samara and variation that we see in the seeds themselves. So what does this actually look like? Well, it allows us to automate quantification of seed morphology really well, and we can quickly assess hundreds of seeds to understand the distribution of morphological trait differences between both the seed and samara, and then relate this to landscape variation that we see. We can relate this to the timing of collection, the year of collection, and more to understand maybe how the environment may be impacting seed production. And this provides us with a lot more information on the standing genetic variation on the landscape today and associated with our ex situ collections. So we did this for all of our seeds, and we were assessing about 50 seeds per maternal family. So what does this actually look like? Well, I'm finally showing you the map of where our seed sources came from. As I said, it covers broadly the distribution of black ash seed that was available at that time from our different partner sources. And you'll notice there are some gaps, and this is just reflects the availability of seed at that time. But if there are opportunities to include more seeds in the future for relatively quick X-ray analysis and morphological assessment of seeds into the future, we think this is a great opportunity to expand upon the knowledge that we have already existing in many of our seed collections. But what is really important here, I think, to take away from this is that when we look at variation across the landscape, we can see substantial variation. About 10% of the variation is distributed within or among populations, meaning that there are population level differences across the range of black ash, and that will impact how we want to collect black ash seeds in the future. It does suggest that there has been the evolution of genetic differences across these populations, and ensuring that we capture those differences reflected across populations will be important. What was really exciting in this, and you can see this in the light green maternal family variation, is that there is a lot of variation within a population among those maternal seed sources within a population. So that tells us that there is a lot of genetic variation in black ash. In when you're out there in a stand collecting seed, if you collect from quite a few mothers, you will capture quite a bit of the genetic variation within that particular population. So it tells us that there is a lot of genetic variation probably within black ash populations, but there is also population differences across the species range. And finally, the great advantage of doing a study like this is we asked for about 100 seeds from our partners. 50 of those seeds were X-rayed, 50 of those seeds went into stratification and germination. And so those seed lots today reflect over 9,000 seedlings from 49 different populations that are they're no longer in our greenhouse. They are overwintering outside right now, but they will be planted in Cone Garden experiments in Quebec, in New York, and Pennsylvania this spring. We're really excited to put these seedlings in the ground, and they do reflect that range-wide genetic variation so we can understand the evolution of trait differences associated with these populations in different environments. And we've been monitoring these populations really closely as they have germinated across time. So we planted them in early May last summer, led by the amazing Kirilo Piccolo. And we've been monitoring the timing to emergence, the timing to development of cotyledons, the timing to development of true leaves, and even the timing to development of the very first compound leaf. And what you can still see in this study, well, the greenhouse, maybe they look fairly similar. You can look at that half-sibling group of individuals from Ontario on the right and note that there is substantial phenotypic variation that we see within our populations from the same maternal seed source. So the indications from the morphological assessment within the seeds is being also recapitulated in our phenotypic monitoring for traits that are important to adaptation, important to persistence on the landscape today. So we're really excited that these 9,000 seedlings will be going out to live in the ground at multiple different environments this summer. And then if I can close off with a little plug that where we can combine our ex situ seed collections with genomic data, this provides us a really great advantage to creating information that is needed for pre-breeding, conservation, and for restoration. We've been using landscape genomics as a tool to inform conservation and restoration of black ash. And we've been doing this, in fact, doing range-wide genomic assessments for Fraxinus nigra, Fraxinus latifolia on the West Coast, Fraxinus americana, and Fraxinus pennsylvanica. And if you're interested in seeing the first publication of our landscape genomics work, it's available online for Fraxinus latifolia, led by amazing former postdoc Anthony Melton. And what we're seeing is that landscape genomics provides the data that we need to be proactive with conservation, including identifying those regional differences that may be important for expanding our ex situ collections, but also allows us to establish more genealogical resources that will be foundational to the species for restoration long term. And so if we think about this, if the goal is to maintain evolutionary potential of our species, both for now and into the future, considering changing abiotic and biotic pressures, combining that information that we collect when we're out there collecting our seeds. We collect information on the provenance, where it came from. We collect morphological characteristics to denote what species it is. Combining that with leaf collections that we can use for a genomic analysis will allow us to create these pre-breeding conservation and restoration resources to understand that genetic variation on the landscape today. And that doesn't necessarily mean you have to have the funds to sequence individuals on the landscape today, but where you combine your seed collections with leaf collections, that will be available from now and into the future. And with that information, we can understand genetic structure, we can understand isolation by distance, how genetic differences are parsed across a landscape, and we can understand current genotype environment relationships. So maybe we can predict and project how we can use that understanding of genetic variation and environmental variation to improve restorations into the future. So ultimately, maybe I've convinced you that this morphological monitoring within our ex situ seed collections can be a valuable foundational resource to understand what we currently have and to allow us to plan for the future. And where we can combine seed collections with leaf collections, we can enable genetic monitoring now or into the future. And ultimately, this can improve the effectiveness of preserving the evolutionary potential of our ash populations, which will be a legacy for generations to come. With that, I'd like to thank all those funders and people that provided us seeds. We couldn't do this without a lot of partners. And so big shout out to the many partners that contributed to this work along the way. Thanks.
Thank you so much, Jill. It's like an immense effort, a lot of people. Thanks for sharing that with us. All right, we're going to shift to our last speaker, and then we'll have time for Q&A. So our final speaker is Jeff Carstens, horticulturalist at the USDA North Central Regional Plant Introduction Station. This is one of the nation's leading facilities to conserve genetically diverse plant materials in ways that can help bring forth new plant varieties and breed resistance to pests like EAB. Jeff is a steward of these critical plant collections, working with a team of scientists to expand the diversity of plant genetic resource collections. He brings extensive knowledge of the station's efforts to preserve ash and allow for its reintroduction once resistant trees are developed. Jeff, take it away.
All right, well, thank you for the invite and introduction. I'm excited to see some of the other talks already, which is kind of a lead way into some of the work that I've been involved with. But basically today I'm just going to kind of give you guys a good overview of the preservation of Fraxinus through seed banking. So I technically work for an organization that's called the National Plant Germplasm System. We've been around, our station opened our doors back in 1948. And so we've been tasked with preserving plant genetic diversity. And so if you looked at any of these sites across the US, they're doing the same thing, just with different taxa. And so Ames got designated as the Fraxinus repository a long time ago. And if you would have looked at our holdings pre-EAB, it was probably less than 3 or 4% of our entire collection. And now, as you can imagine, with all the work that's happened post introduction of EAB, our collection is almost close to 20% ash. So our mission here at the NPGS is to conserve genetically diverse plant germplasm and the associated information. So we're doing that by depositing seeds into jars, packets. We're growing out plants into our fields to collect seeds off of those. And we use controlled pollinations or isolations to try to get increases so that we can have materials available for distributions. We're even doing biogenic preservation of dormant budwood, and which is really a labor-intensive activity where you're basically collecting scions or cuttings in the dormant season. And there's a very intensive process of getting those prepared before they're going into liquid nitrogen. I think all the speakers have already mentioned how it requires a interdisciplinary approach, and there's a lot of collaborators involved here. Showing you a picture of some of the activities that encompass this, and largely it's starting with the collections, making those acquisitions out in the field. And this is no easy task. Jessica talked about the efforts to do seed sourcing, and it does take a lot of effort and skill. Once we get those samples, they get viability tested, typically through a TZ test, and sometimes it's just a visual test. The seed samples, once they're stored, we look at the quantity and the quality of those and determine if they can be made available and distributed. So that's our major purpose here, is to have these materials available for research, just like what Jill talked about with her work. And if we didn't have these in storage, it would be, I couldn't even imagine how we could get the research done without having access to these samples. The project goals, regardless if it's Fraxinus or any other species that I'm working with, is to try to cover the species' entire native range. And that includes also the taxonomic diversity. So if we look at Fraxinus as an example, we currently have about 36 species out of 70 in our holdings. So we have a long ways to go to trying to get all that representation. But I did like one of Jill's comments about the temporal sampling, is that, yes, we are out there making collections. Other people are out there making these collections, and it's a one-time thing. And kind of the next thing I think perhaps on the horizon is going back to some of these sites and doing additional collections once the understory seedlings hopefully get to seed-bearing age. While we're doing all these collections, the main goal is to collect good passport data. And the passport data is essentially, where did it get collected? Using a GPS, taking coordinates for every sample that's collected, making notes about what the habitat was like, the size of the specimen, perhaps phenology information about, the tree was in folk color or wasn't. And we're taking herbarium specimens and images while we're out in the field, and all of this data is getting documented into our database. While we're out in the field, we're trying to optimize a sampleizing strategy to try to capture that 95% genetic diversity of a population. So we're looking at trying to collect seeds from specimens that are young, middle-aged, and old, and then looking at perhaps differences in morphology of the leaves or the seeds or the phenology of the plants to try to potentially guess that we're capturing that genetic diversity. Everything that I try to do and try to influence is when we're making out these collections is population-based sampling, and I'll talk a little bit more about that here shortly. Now, all of this work involves the help of other partners. So the U.S. Forest Service has been huge in providing and getting out there and making collections. We have a lot of private folks, landowners that are interested in doing this. The tribal groups have been very key in making these collections. Some of you may know Les Benedict, who's been, from the early days of making collections and getting these things deposited, is essentially resulting in a legacy that will provide plant genetic diversity for the future generations. So I mentioned the documentation. All of this information is available on our Green Global database, which includes the passport data of where it was collected and the associated species, how many specimens were sampled, and it indicates our current availability. So if a researcher wanted to request some samples, they can search on, say, just samples from Iowa or just samples from New York, and select those that are available and putting it in essentially in a shopping cart, if you will, and request those. So we just finished a recent trip down in Arizona near the Grand Canyon. And just wanted to show you guys an example of what we're dealing with, and because as Jessica mentioned, and I can't reiterate this enough, is like the efforts that it takes to get into acquiring these is very time consuming and labor intensive. What's fun about this picture, it kind of gives you an example of the vastness of some of these areas. The other thing that you might notice is if you look at the blotches of a little fall color of yellow, those are all ash. So these are Fraxinus anomalous and Fraxinus cuspidatus that are scattered across the canyon here in Arizona. And what you might notice is, like, if you look at this picture from this angle, you can see all of the yellow. And so these are slight north-facing slopes where it's just cool enough for the Fraxinus to be present and persist. If I was to walk a couple miles down the canyon and take this image the other direction, you may not see any Fraxinus at all because they're just on that downside ridge of where that north-facing slope is. So that, raises questions about, like, when we're doing restoration efforts, of like how important it is to really know your site and to really strategically place plants into their appropriate areas so that they can thrive and persist. So when we were down here in the Sycamore Canyon, that population of Fraxinus cuspidata, it was assigned an accession number of 36291, and I've indicating here a line showing the distance between the two samples that were the greatest distance apart, and that was almost a half a mile. But you notice there was a concentration of specimens in one area. That's where we were able to find seed. This next image is kind of backing out just a little bit, and this far dot off to the right is where we parked our car. So it was probably close to a 4-mile hike to get to this population, and it literally took all day to get those. So again, mentioning the efforts that it takes to get these samples. Once it's all collected and cleaned, we will assign these an inventory number. The accession number, that 36291, is kind of that, what we call the family or the house, if you will, and all of the children that were collected and sampled get a special inventory number that tells us also where it was located. And you can see, like, some trees are really productive, some trees aren't. And sometimes we just have to take what we can get if we don't have enough plants to sample. So from there, once these seeds are put in storage, we have opportunities to kind of look at viability. And sometimes that's looking at viability over time. And so when we first started working with the storage of Fraxinus, we had placed split samples in the refrigerator at 4C and also in the freezer at -18C. And these are side-by-side comparisons of 15 years after storage. We can see that materials in 4C are basically at or near dead. And so we are providing recommendations to others that are trying to go out and collect seeds to do not put those in the refrigerator. But if you put them in the freezer, they're good. Seeds are living organisms. They're not going to last forever. And I guess, I can mention that one of the oldest samples that I have was blue ash that I think it was collected back in the early 80s. And we did a germination test, I think it was like 2015, and those seeds were perfectly viable. So after so many years in storage, we know that keeping them in the freezer is a good option. Moving forward, so we've kind of done a lot of collections across the eastern half of the U.S., focused on what I consider the main four. So black ash, blue ash, white ash, and green ash. Now we're starting to shift over to some of the southwestern species. And, over the last couple of years, the Oregon Department of Forestry had made a lot of collections of Fraxinus latifolia. So you can see in the table over there that there's a total of 93 accessions that we have. And I should clarify that, an accession in our system may mean numerous samples within one accession. So if we were to total up how many samples we have in storage, it probably would be around 6,000. There are still numerous taxonomic and geographic gaps, as Jill pointed out. So there's still work to be done. And then, of course, I should mention the lingering Fraxinus genotypes. If folks are out there seeing something unusual, being observant, and there's opportunities to collect seeds from those are highly interested and sought out samples that we would like to have in the seed vault. Finally, help is always needed. There is no way that this can be a single organization or institution. It requires help from all institutions, all organizations, and as many people as possible. I'm always interested in people that are wanting to make collections and deposit seed samples, whether they're going to be used right away or used 50 years from now. Those samples that people are making will have a long-lasting use into the future. In addition to people getting out there collecting seeds, sometimes we just need information from people locally of what is the seed production happening with right now. As an example, in 2014, my team was trying to target blue ash collections in Tennessee and Kentucky. And for almost 5 years, there was absolutely no seed production on ash and the blue ash. And so we didn't make any efforts to collect samples until finally we got information from locals that said, hey, it's a really good bumper year for seed production. And that's when we went out and we made those collections. And also, of course, we need help knowing where the hot spots are. Where can we find ash? I would say for a lot of these eastern species, they're relatively common, but it's a whole new ball game that we've run into trying to target some of these southwestern species. Is they're not easy to find, and they're definitely not easy to get to. So that's where we really need help on just knowing exactly where to go. So with that, I appreciate your attention and everyone being my captive audience for today. And if anybody wants to reach out to me at a later time, this is my email. Happy to discuss plans moving forward if there's anyone interested in making seed collections and depositing those into the seed vault. So thank you.
Great. Thank you so much, Jeff. Thanks to all our speakers. So I want to open it up at this point to questions and discussion from the group. You can put questions in the chat, raise your hand, you can unmute. I can get us started on this section. I have a question for Jessica. Could you tell us a little bit more about the status of reforestation efforts for Raspitha tribe and kind of how you select for seeds? Are you selecting only basket quality seeds? Are you selecting for genetic resistance? What's your approach?
So it's a little early to select for genetic resistance, but we're really opportunistic seed collectors. So we're not even focusing specifically on black ash. If we find an ash tree and it is flowering and there's going to be a lot of seeds, we're going to collect those seeds. But it is a little soon to get into the resistant specifying EAB resistant trees, but it is something we're going to keep our eyes on in the future.
I had a quick question for Jessica on the harvesting of the black ash for the baskets. Is there a specific time of year that those trees have to be harvested to get good quality strips, or could those be targeted at certain times of the year, specifically the time when seeds are ready to be collected? So you could do a kind of a two birds in one stone kind of a thing with collecting seeds.
So we don't normally harvest the seeds that are with the trees that are seeding because we want the seeds. But the actual harvesting, it can happen at any point of the year. But we do a lot of them during the winter because we want to do as little ground disturbing as we can. So we normally will go out in the fall, we collect the seeds, we target the ones that we think are going to, we should harvest for baskets. And then we return in the winter, this time of year specifically, when the ground is nice and frozen, and then we harvest.
Thank you.
We had a question earlier in the chat about whether you could harvest dead standing ash for basketry.
That's actually something that we talked to a lot about with Nate Seigert, and whether or not that's something that we want to start looking into. Because right now we're really not sure. We know that if it's early in its infestation, that there's still a lot of viable tissue in there that could still become splint. But we don't normally remove dead standing just because there wouldn't be a lot of viable tissue under the bark. And it is labor intensive to harvest a tree and drag it out of the forest just to find out that there's no viability.
We had a question just come in from Kathy Rooney. Can folks doing restoration projects plant nursery seedlings now, or should they wait a few more years?
That might be a good question for Jill.
I mean, I think the material that you might get from the nursery today won't be the material that has been assessed to be resistant or lingering with respect to EAB. So there's a reasonable chance that when it gets to a particular DBH, it could be susceptible. We just don't know enough about it right now. I think it's still good to get it on the landscape and to have it as part of the landscape today, though, because it does provide a lot of services. So yeah.
I'll add to that as well that it really depends on where you are in your area and what your EAB infestation looks like right now. Because on our timeline, we believe that if we were to start producing seedlings today, that by the time they reach susceptible DBH, we will be at our peak infestation, and that it's just not ideal timing for us right now.
One of the things that I've been really interested in is the degree to which the stump sprouts can be used to maybe as seed producers. So when we've seen this around this region, particularly for green ash, and maybe this is a question for Nate and Jessica and others. What is the value of the seeds being produced from stump sprouts for collections?
Yeah, I think that's a good question. On one hand, they probably don't have a lot of genetic tolerance to emerald ash borer, but they did have the, capacity to produce viable stump sprouts that made it to seed producing size. So that there's probably some value in that. I think it'd be interesting to keep that, those seeds kind of separate. Jeff mentioned, seeds from lingering ash having a lot of value. And I think you're right. I think maybe there's value in seeds from stump sprouts as well, just, as another tool or mechanism that we can have moving forward.
We had a question in the chat from Jill. No, from Tina to Jill, saying that it was a tremendous effort to see all the work that you've been doing with X-rays to access genetic components over time and the environment. Can you give insights into how this genetic variation might have evolved over time or to climate change? And I think what she's getting at is it possible to kind of cultivate a super resilient ash tree with this knowledge?
So I can tell you from some preliminary analysis so far, that it seems to be that there is indications that climate is driving morphological variation in the ash seeds, and we can see evidence when we control for year of collection and geography that as from the coast to the interior, there's a gradient there in precipitation that appears to be contributing to differences in morphology, as you would expect. And there is also a latitudinal gradient in temperature that you would kind of expect to be contributing to variation in seed morphology. Um, can this give insights into so I think that it tells us that climate is impacting the evolutionary trajectory of these populations, and we're seeing that in the morphology, and we're actually seeing it in the germination and the traits and how the seeds are developing at a very early stage. Um, can we cultivate super resilience ash? I think there I would be more cautious because one of the things we could maybe do something like that, but then what if something changes and then that resilience is completely lost? And the goal, I think, with the seed collections into ensuring that genetic diversity is preserved is to kind of hedge your bets, to have that diversity as we experience new abiotic and biotic threats.
Question just came in from John Irwin. How much sapwood should harvested logs have to make them useful for basket making?
Oh, sorry, I was responding to another question in the chat.
It was asking how much sapwood do you need in a harvested log for it to be used in basket making?
So in general, so when we harvest the log, you can, when you do that pounding and you go around, we can go around, depending on the size of the tree, usually about 3 times. And then from there, we normally have enough splint to create about 3 baskets, but obviously it depends on the size of basket, type of basket. If you're making the bigger utility baskets, it's obviously going to take a lot more splint than if you're making smaller fancy baskets.
All right, we're going to have one final question and then we'll close things out. Question from Chad Fieros. Jill, how did you choose your sites in Quebec, New York, and PA for planting the seedlings?
That's a good question. This is a lot of work, putting in these large scale provenance trials. We do this a lot in forestry, and we really rely on partners. So one of our goals was to really have all the diversity of the ash seed in the core of the species range, which in Quebec, the plantation will be just outside of Quebec City, near Laval University, with our partners with the Canadian Forest Service. That is kind of a large core of the species range. And then we wanted to, because we are also interested in the long-term resilience of black ash to both the emerald ash borer and climate change, we wanted to ensure that we were going south as well. So the New York site is still, which will be near SUNY ESF, in near a lot of chestnut plantations where people have a lot of experience with these type of plantations. So we'll leverage that climate, which is still within the range, but quite different from the Laval climate. And then the Pennsylvania site is more on the periphery of the range, so that would be like a climate forward site. So assessing the traits of the populations here might tell us how the populations will do under a more warming climate. So that was kind of the strategy, and then having amazing partners at those different locations was vital to planning.
Well, I want to thank our speakers again for giving their amazing talks. Thank you so much. Thanks to our participants for coming. Just a reminder, we've got another event coming up in 2 weeks on March 6th. It's about the kind of the how-to of ash seed collection. So if you want to take some of what you learned today and take it into the field and start trying to collect some seed, that's what that next event is about. I'm going to be sending out a short survey for you all to share feedback on the event. This kind of feedback is really helpful as we build out additional events for the network. So please, if you have time, it takes about 5 minutes, fill out the survey. Thank you all. A recording will be sent shortly. Hi, everyone.
Okay, thanks, everyone.
Thank you.
Thank you.
Thanks.